our last sperm talk, and now we're going to learn about sperm motility from Daniela Chavez.
Despite my efforts to wear 4-inch heels, I still get the booster chair. Okay, hello everyone. Let's make sure this is working. My name is Daniela Chavez. I am also a graduate student in the Stanfield Lab at the University of Utah, and I'm going to talk to you about later in spermatogenesis, the process by which C. elegans sperm gain motility. So for sperm cells, cues in the extracellular environment are critical to their ability to fertilize oocytes. They rely on these extracellular cues to undergo the final steps of spermatogenesis, which are required for migration and fertilization. These cues include seminal fluid as well as components of the male and female reproductive tract. With my work, I'm interested in understanding factors that contribute to gamete success, and so today I'll tell you about a serine protease inhibitor called SWM-1 that regulates sperm motility. Within the male, sperm remain immotile. Upon mating and receipt of extracellular cues, they become polarized cells with an extended pseudopod that they use to crawl towards oocytes. In this activated state, they are now fertilization competent. We know that the timing of activation is very critical to fertility because various genetic mutants in which males contain prematurely activated sperm within their seminal vesicle or the sperm storage organ are infertile. One such mutant is the SWM-1 mutant. When dissected out, as is shown here in this image, SWM-1 mutants have prematurely activated sperm with a fully extended pseudopod. This causes a significant reduction in fertility because SWM-1 mutant sperm cannot be transferred to hermaphrodites efficiently, indicating that the role of SWM-1 is to inhibit activation. Now, downstream of SWM-1 is a protease Try5, which is a seminal fluid protein that's transferred to hermaphrodites during mating, which is shown here in a still of a movie where a Try5 GFP labeled male has mated with a hermaphrodite, and we can see Try5 GFP expanding throughout the uterus of the hermaphrodite. So this and other data has shown us that Try5 acts as the signal that triggers activation. But exactly the molecular details of how SWM-1 inhibits activation has been unclear. But studying SWM-1 also gives us an opportunity to learn more about this specific class of protease inhibitors for which little is known. So SWM-1 has two trypsin inhibitor-like domains, or TIL domains. TIL domains can be found in proteins of Drosophila seminal fluid, frog skin secretions, and are also components of larger proteins such as von Willebrand factor. And mutations in the TIL domains of von Willebrand factor are causative of this blood clotting disorder. So studying how SWM-1 inhibits activation provides us with an opportunity to learn mechanisms of regulating cell motility and factors that contribute to fertility. But it also may inform us about regulation of proteolysis in other biological contexts. So to understand how SWM-1 spatially and temporally regulates activation, I first wanted to know what cells in what cells is SWM-1 expressed. Because in addition to the two TIL domains, SWM-1 has a secretion signal. So I wanted to know what cells might secrete it. To do this, I generated a transcriptional reporter where I fused the promoter of SWM-1 to mCherry and a histone protein. So what we're looking at are the nuclei of cells that express SWM-1. I found that SWM-1 is expressed in the posterior of the male, specifically in a subset of vas deferens cuboidal cells. So the vas deferens essentially forms a tube that secretes seminal fluid and through which sperm pass during mating. In addition to vas deferens cells, SWM-1 is expressed in extragonadal cells in the posterior of the male. And I confirmed through colocalization that these are actually muscle cells. These SWM-1 expressing muscle cells extend about halfway up the seminal vesicle. But remember, SWM-1 is a secreted protein, so I also wanted to know where SWM-1 protein localizes. So to do this, I generated a SWM-1 mCherry fusion and knocked it into the endogenous SWM-1 locus using CRISPR. This generates a fully functional protein that's capable of inhibiting activation. Not surprisingly, and in line with the transcriptional reporter data, SWM-1 localizes to large secretory vesicles in the vas deferens. and in a subset of these vas deferens cells. The subcellular localization within these cells is very distinct, where the largest vesicles are aligned to the apical membrane as though they are poised to be secreted as seminal fluid. And I did confirm in an independent experiment that indeed SWM-1 is a seminal fluid protein that's transferred to hermaphrodites during mating. Additionally, and strikingly, SWM-1 is excluded from the most posterior vas deferens cells. And so, like many other systems, seminal fluid in C. elegans is a combination of secretions from various tissues, which include the valve and distinct regions of the vas deferens. In addition to localizing to the vas deferens, and in line with its role as an inhibitor of activation, I found that SWM-1 is present near sperm. Here we're looking at the seminal vesicle, and we can see that SWM-1 localizes in pools that surround developing spermatids. And it's also present faintly in the seminal vesicle. And also, lastly, SWM-1 is a secreted protein, and I see that it is present within the pseudocoelom and can see that celomocytes take it up. So what I've shown you is that SWM-1 is expressed in the male, the posterior of the male in muscle cells, and it's also expressed in the vas deferens. The protein localizes not only to the vas deferens, but it also localizes to the seminal vesicle. And since I'm interested in understanding activation, I wanted to know the source of SWM-1 or what tissues are producing SWM-1 that goes to the seminal vesicle. So to do this, I did a tissue-specific expression of SWM-1 under either a muscle-specific promoter or a vas deferens-specific promoter. So we're essentially looking at a sufficiency experiment where I've quantified the sperm activation phenotype, and I'll walk you through this. Importantly, all of these transgenes, the muscle and vas deferens specific, are in a SWM-1 null background. So on the y-axis, we have the percent of males that contain either non-activated sperm, a mix of sperm and spermatids, or in black, activated sperm. So the controls are wild type, contain non-activated sperm, and then SWM-1 mutants activated sperm. And also, I generated both tagged and non-tagged versions of each of these transgenes so I could determine protein localization. So I'll first draw your attention to the vas deferens derived SWM-1. We can see that vas deferens derived SWM-1 does rescue activation. However, it is not sufficient because if we look later at 48 hours post L4 in adulthood, it no longer rescues. And much to our surprise, SWM-1 derived from the muscle is sufficient to rescue activation. And consistent with this, muscle SWM-1 M. cherry does localize to the seminal vesicle and surrounds developing spermatids, whereas vas deferens derived SWM-1 is not detectable in the seminal vesicle. We do believe that the rescue we see at this time point is due to SWM-1, and perhaps it's at levels too low to detect at this time point. So this leads us to a model where we believe we've identified this somatic signal that's signaling to the germline, where SWM-1 is secreted into the pseudocoelomic cavity where it can be taken up by the gonad and inhibit activation. And what you couldn't see from the movie or from the image I showed you of Tri5 is that we also know it's present in the seminal vesicle in line with this model. But I also showed you that SWM-1 is a seminal fluid protein. It's present in the vas deferens. So in addition to its role as an inhibitor of activation, it seems that perhaps it may have an additional role as a seminal fluid protein and could inhibit another protease within the hermaphrodite once transferred. It turns out that at the molecular level, the idea that SWM-1 could be a dual functioning protein seems likely. So if you remember, SWM-1 has 2 TIL domains. These domains have 10 conserved cysteine residues, as well as a 3 to 4 amino acid reactive site that, where the protein is predicted to inhibit the protease. And they have been shown biochemically by others that they have protease inhibitory activity. The TIL domains of SWM-1 are actually different from each other. There's the N-terminal TIL domain and the C-terminal TIL domain, leading us to the hypothesis that perhaps SWM-1 could inhibit 2 distinct proteases. So to test this in the context of activation, I generated single TIL domain containing transgenes, and I again placed them in a SWM-1 null background. So here I am showing you again a quantification of the sperm activation phenotype. I've just turned the graph on its side. On the left are the genotypes, and then I've also drawn them out schematically here. So in the first column is the endogenous SWM-1 locus, and then in the last column is whether or not I've included a transgene. These first three are all controls, so we have wild type, SWM-1 mutants, and then importantly, a full-length transgene that includes both TIL domains is sufficient to rescue activation. However, we can see that the two TIL domains are indeed not equivalent. So the N-terminal TIL domain does not rescue activation, and the C-terminal TIL domain alone partially rescues activation, indicating that the two TIL domains are not equivalent, and that it seems that with respect to activation, the C-terminal TIL domain plays a more predominant role when it comes to inhibiting activation. All right, so what I've told you is that SWM-1 is a somatic signal that is signaling to the germline. We believe that it is being secreted by muscle cells into the pseudocoelomic space where it is taken up by the gonad. I'm currently doing experiments to test exactly if this is specific to SWM-1 mCherry, but I'm very curious to understand exactly how this is happening at a cellular level, how it might actually be getting into the gonad. So if people have any good ideas, I'd be very excited to talk to you. And then lastly, I told you that SWM-1 is this 2-TIL domain protein, and that the TIL domains have non-equivalent roles when it comes to inhibiting activation. Whether this means that there's another additional protease in the pathway is a possibility, but it's also fun to speculate that perhaps this additional role of the other TIL domain is important for a post-mating role. And with that, I would like to thank you all for your attention. I'd like to thank my lab, especially my extremely supportive PI, the University of Utah Cell Imaging Corps, the Phillips Lab for their excellent advice when it came to generating the knock-in, and as well as the organizers for giving me this opportunity to speak. I'd like to take any questions.
Does SWM-1 have to come from the muscle, or can you ectopically express it in some other somatic tissue and get it to signal?
Other than expressing it from muscle and vas deferens, those are the only places that I've expressed it from. So I imagine that it's possible, but those are the only ones I've tried.
So for the TIL domains, is it just better to have 2 TIL domains? Are there differences in sequence between the N and C, and could you switch them or use them like 2 similar ones together to see if there's placement issues?
Yeah, we haven't just done those experiments precisely. The domains themselves are very small, but they are extremely similar to each other.
So I might have missed this in an earlier paper, but did anyone ever make just a pure transcriptional fusion to see which cells normally are the source of SWM-1 expression?
Yeah, so that was earlier. Do I have a second to go back? Does anyone care? Yeah, so it is expressed in muscle cells as well as vas deferens.
Any other questions? Oh, yep. Yeah, sure.
If you take your muscle-specific line that's rescued
Mm-hmm.
and made it to a hermaphrodite that's null, do you get any phenotype that would reveal some requirement for that vas deferens source?
Yeah, so I have not specifically done that experiment. Certainly not with the muscle, but I am working on doing some sperm competition experiments. I think I can also be a little bit more specific with regards to the seminal fluid, because I can express it only in the vas deferens and ask about its requirement, a post-mating requirement.
Okay, thank you.